Welcome back. Let's take a minute and look at a couple of animations to help us clarify these ideas of passive transport. Here we can see particles moving from high to low. Think about the difference between simple diffusion and facilitated diffusion. Notice over on the left, in simple diffusion, those particles are moving from high to low through the phospholipid bilayer, where on the right, in facilitated diffusion, they're moving from high to low through a protein channel. Both are high to low, both are moving down their concentration gradient, therefore both are great examples of passive transport, not requiring energy. Next, let's think about how substances would diffuse. Here we see those solute particles moving through the membrane from where they were high to where they are low. So there's our diffusion. And now let's think about how that diffusion differs. In the previous example, the substances were simply moving through the phospholipids, but here, these substances needed to move through the channel. This was still high to low, still going with the flow, but this was facilitated diffusion because it was moving through those protein channels. Next, we can see the movement of water from where water's high to where water's low. Remember, movement of water is going to be called osmosis. We've got one more scenario. What's going on here? Everything! The blue substances are moving by diffusion, the red moved through facilitated diffusion, and the water molecules, represented by those sort of pinkish Mickey Mouses, those were moving by osmosis. All three of those passive transport. Remember we said we care about all this cell transport because of how it affects cells. So now we're going to learn how to think about which way water would move in and out of our cells. So we're focusing now on osmosis, thinking about how water will move down water's gradient. When we're given a situation like this, we're drawing this as a beaker or the environment, and then the structure inside is representing a cell. Notice if they're giving us a percentage of water or solvent, we can then make some assumptions about the percentage or concentration of solute. So if the cell is made up of 80% water, we can then infer that that cell must be 20% solute. As we look at the surrounding solution, it says 100% water, so therefore we know that there is no solute. The value in the environment has to add up to 100, and the environment in the cell has to add up to 100. As we look over here, we've got 80% water in the cell, therefore 20% solute in the cell. And over here, 80% water in the cell, therefore 20% solute in the cell. It's nice to have all those values so we can really think about where this water is going to move. There are a couple of different ways to look at this. We said earlier that osmosis is the movement of water down its concentration gradient, meaning moving from where water is high to where water is low. So if we look at that in this situation, we see that the water number is higher in the environment and lower inside the cell. Therefore, water will move from the environment into the cell. We also said that water will move to where all the solute is at. So the water is going to try to get into the cell, because that's where all the solute is, to try to break up some of that solute and balance things out. What about in this next example? Which area has more water? The environment has 80% water, and the inside of the cell has 80% water. It's kind of a trick question. There is no area of higher water. Therefore, if a little bit of water wanders in, a little bit of water will also wander out. And we call that equilibrium. It's pretty much balanced. They're the same amounts of both substances on each side. Now let's look at the third situation. We can think of it a couple ways. Remember, we can say that water moves from where water is high to where water is low. Which water number is bigger? 80 is bigger than 70, so water will move from where water is high to where water is low. We could also think of it, though, as water going to get to where all the solute is at. Notice there is more solute in the environment, so the water's going to try to get there to bust some of it up, to dissolve some of it, to balance these out a little bit. There's some key terms that go along with osmosis that we can use when we're describing relative amounts of solute in the region. So the first way we can look at this is that if the environment has more solutes when compared to the cell, then we can say that the environment is hypertonic to the cell. Just like if you are hyperactive, you have more energy, a hypertonic region has more solute. So we can think of it this way. If this is our beaker, and if this is our cell, and we're saying the environment has more solutes, that's like saying this environment, this beaker, has a whole bunch of stuff dissolved in it. So now that I know where all the solute is at, and I know that water tries to go to where all the solute is at, once I have this picture drawn, I now know that water is very likely to move from where water is high to where water is low, or water's trying to get to where all that solute is. So we can say that if a cell is in a hypertonic environment, that water is going to diffuse out of the cell. And as that happens, as water leaves a cell, the cell is actually going to shrink. The opposite of hypertonic is hypotonic. Hypo, below. And what we mean by that is if the environment has less solute than the cell, then we call it a hypotonic environment. And we can picture that again with our beaker and our cell. But this time I need to show that there's less solute in the surroundings. So I'm going to put some solute in the cell. The environment has less solute, hypo, below. And if I know water wants to get to where all that solute is, water is very likely to try to rush into the cell. And as that happens, the cell was shrinking before, but now if the water's entering, the cell grows. So hypertonic says the environment has more solute. A hypotonic environment has less solute. And the last possibility is if the environment has the same concentration of solute. And in that case, we call it an isotonic. Same amounts of solute outside and inside the cell. To represent this one, I've got my beaker, I've got my cell, and I want about the same distribution of solute inside and out. Notice, since there is not an area with tons of solute, if a little bit of water comes in, a little bit of water wanders out. So really, water is kind of just diffusing in both directions with no overall change. We say there's really no net movement. It's one thing to think about a picture where arrows are moving, but it's another to really think about the impact this movement can have on cells. So first of all, we've got animal cells up here, and we've got plant cells down here. Remember, the big difference is going to be that animal cells are only surrounded by a membrane, while plant cells have a membrane and a cell wall for some extra structure and support. So here we've got a cell, and it has been placed in a hypotonic environment, meaning there's less solute surrounding the cell, therefore more solute inside of the cell. Hypo, below, less solute out here, which is why water rushes in to get to where all that solute is at. And depending on the type of cell, we can call the effect two different things. In an animal cell, if water keeps rushing in, the animal cell could actually burst. However, in a plant cell, the cell wall prevents bursting, and we say that those plant cells are turgid. Next up shows the impact of cells soaking in an isotonic environment. And that just tells us that we've got the same concentration of solute inside and outside, which means if a little water wanders in, a little water also wanders out. And this is going to be the most normal for an animal cell. If this happens in a plant cell, it's not going to be quite as plump and full and happy as it was over here. Instead, it's going to be a tiny bit limp, and we call that flaccid. The final situation shows if we've placed our cells in a hypertonic environment, meaning that there's more solute in the cell's surroundings as compared to the inside of the cell. Water tries to go to where all that solute is at, which tells us why that water is leaving the cells. In an animal cell, we can simply call this shriveled. And in the plant cell, the membrane is actually going to shrink in away from the wall. And we can describe this as plasmolysis. These are a lot of new words, but just think about what they mean. Hypo, below, less solute in the environment. Hyper, more solute in the environment. These words are always referring to how much solute is in the environment. So this chart will definitely help you thinking about the impact on plant and animal cells. Your next job is go ahead and tackle this flow chart. But notice that today we only covered the passive transport side of things. So I want you to go ahead and complete this left-hand side of the flow chart today. And you've got one other activity that you're going to work on. Thanks so much for all your hard work on the cell membrane and cell transport. We'll do a lab next class, so dress smart, and we'll get to see all of these great properties at work.